I can start. Thank you so much for inviting me, Dr. Dawson, Dr. Murphy. I will be talking a little bit more into the details of the molecules, what's happening in the vision research. And I would like to say that 15 years is a very long time to predict. I think even 5 years is very hard to predict. 5 years ago, as you can see, the whole genome sequence was not imaginable to sequence in just 24 hours. Nobody would believe that. And transfection of just 4 genes can reset a cell to pluripotent stage, like the iPS cells. And also there's a technology now that you can cut and replace genome sequence at the location of your choice, and then developing knock-in mouse animal models. And there's another technology with the virus vectors, which can penetrate through the entire retina. All these technologies is now called in these names. But these were not available 5 years ago. So predicting 15 years ago is a little bit difficult for me. But the goal for these research has been the same for the past 30 years or so. And there's only 2 goals. One is quickly, accurately, and cost-effectively identify the gene of the disease-causing mutations. And this has been the goal for many, many years. But now it's gradually coming with a collection of data. And I have to emphasize that the collection of data for all this mutation is very important, because once you have all the information, then you don't have to sequence the entire genome to find the mutation. If you have the selective mutation in the database, you can only look at those mutations only. And also these instruments to do the genetic testing is available currently, and I don't think that's going to be much of a problem in the future. Also, once you find the mutation, how are you going to cure all these genetic diseases? Now, there's a lot of work that's being done, but there's a problem for every procedures that is being, or research going on. One is gene therapy. Gene therapy has been going on for 30 years, and gradually now it's coming into the vision research. Also, drug therapy. This has not been going on too much. And this is, I will go into the details later, that to develop a drug, you need to identify what the mutation is actually doing. And sometimes it's very difficult to predict that the mutation is going to do in your body. And also, there's so many mutations that's found. You cannot invent a drug for each mutation. So you have to sort of do a research that all these diseases can sort of come to a common pathway that you can cure with 1 or 2 drugs in the future. And that will be a very audacious goal for drug therapy. And also replacement therapy, and this can be a cell, a tissue, and this usually we are thinking about the iPS cells that comes from the patients. You convert to a stem cell stage, and then from here you can differentiate into sometimes optic cup, photoreceptors, RP cells, retinal ganglion cells, and then you replace it with the bad ones in the patient. So it's a transplantation to the patient's eye. And of course we have been known about the artificial, the lens, the cornea, and of course the artificial, the retina. So I'd like to start with the collection of data about the global collection of genetic data. As we know that the Homo sapiens has migrated from Africa for tens of thousands of years, and then for each individual we have 20,000 to 30,000 variants in our nucleotides. As a total, that's huge numbers of this. However, the sequence cost has been dramatically going down, and then we can sequence the entire genome in 24 hours, almost to the $1,000 levels. Probably by the end of this year, we will come to that goal of $1,000 genome sequencing. What kind of variation are we talking about? Most of the variations are single nucleotide. I don't have the pointer, but single nucleotide variations, small variations, and small insertions. And for these sequences, the variations, we can use the technology called the whole exome sequence, where you can trap the genome and then you sequence with the next generation sequencers. However, to identify large deletions, large insertions, inversions, duplications, and also chromosomal translocations, you need a technology called the whole genome sequencing, which is coming down, but it's still very expensive. Now, all this mutation resides in this category. At the x-axis is the frequency of the mutations. So for AMD, glaucoma, cataracts, and myopia, these are relatively common variants that we see. So you see these variants in patients and also in the controls, normal people as well. However, if the frequency comes down significantly low, you will find a Mendelian diseases, which is RP, Stargardt, macular dystrophy, cone dystrophies, and et cetera, et cetera, et cetera. So these Mendelian diseases is a recessive form, a homozygous recessive form, compound heterozygous, or dominant form. And for exome sequencing, you have to sequence all of these patients, parents and siblings. But for the dominant, you have to sequence all 3 generations. So that's very costly to identify. That's why we need a big database so that you don't have to sequence for each pedigree we face the whole genome to identify the genes. So this is the Mendelian retinal diseases that we usually see in the hospitals. And then this is the sort of the mapping of the genes since 1980, and then go the way, and then the gene that has been identified. What's going to happen in the future probably is that we're going to be stopped doing all the mapping, and we're just going to do the sequencing. And because of this technology, we will probably quickly identify all the genes in the next 5 to 6 years. And it will plateau. And then we can begin to develop the complete sort of variation database for do a quick diagnostic. So this is all the gene for the retinal diseases. We have experienced through our research in Japan. I am in here. We have all the 25 universities collaboration, sending all the samples to us, all the clinical information come to us. We send the sequence to RECAN. The National Genetic Institute will do all the analysis, comes back, and feedback, and we do the drug development. And we have tackled 35 diseases using all these instruments. But we have now know that only 17% of the pedigree that we sequenced has the known mutation that has been published. So that means the huge pie of this percentage is still unknown for the Japanese population. I'm sure this is going to be the same as the Indian population, same as the Chinese population, same as the Philippine populations. So right now, these are the 2 regions that are leading all this genetics, but we have to catch up, and then the Asia, the South America, and the Africa will probably, using this technology, will quickly capture and then make a good database. Also, the genetic testing, new devices coming up, like the nanopore technology. This is the size of a USB stick. And then you just isolate the DNA, put it into this device. It has thousands of pores, nanopores, and the DNA goes through the pores, A, C, G, T, it detects, something like this. So you can easily, these kind of things will be used to do once we have the database, you can have a USB stick to sequence what the patient will be, what kind of mutation he has. Also, currently we are using this technology, currently in Japan, with one single tube, you will have about 96 mutations detectable, and you can sort of sort it out with this instrument to find the mutation very quickly and very cheaply. So what about the therapies? Gene therapies. Currently, there is a therapy for Leber congenital amaurosis, RP65 genes, and also choroideremia has been done, and retinoschisis coming up. So there's many gene therapy trials going up, but very good news is that there is a vector called 7M8. This 7M8 is an adeno-associated virus, but it has a different, they modify the coat of the virus so that this usually the AAV virus cannot penetrate through the retina. It stops at the retinal ganglia cells. But because the coat is different, it can go all the way down to the photoreceptors. And this can be applied to many diseases of the retinal diseases. How about the drug development? We have been working on the normal tension glaucoma in Japanese population. We especially focused on the gene called optineurin, E50K mutation. We made the iPS cell from these patients and controls within the families. We differentiate this iPS to a neuronal cell through these protocols, which I will not speak in detail today. But once we have this sort of pseudo patient neuronal cells of the glaucoma patient, here is green dot is the normal optineurin. It's spread out. This is the nucleus. This is the ER, and it's spread out equally. However, if you have a mutation, it's precipitated in the ER. So we tried to identify what the cause, why is it precipitating? We soon identified that this mutant is interacting with a completely different protein called the TBK1. Usually, the normal optineurin can only interact with optineurin, but this mutant is TBK1. So this is going to be a unpredictable, very difficult to predict. So what we did is that we got the TBK1 inhibitor for this BX795. This is the inhibitor for TBK1, and applied to this the cells. And usually it's precipitated, but you increase the dose by tenfold. You can see that this precipitation is going away. And these kind of, each mutation requires a sort of a detailed research, but if you do this, you will come to some kind of a solution. So this is another example that we have been doing. This is a sustability gene for AMD. We know that there's a two type, the dry type with a drusen and geographic atrophy versus the choroidal neovascularization. And this is the typical in the Caucasian, while this is the typical in the Japanese population. From all the gene analysis, this is the chromosome 1 to 22 here, and this is the P values. And you see that there's two regions, one in the chromosome 1, where the complement factor 8 gene resides. There's a strong association, P minus 1 minus 283, versus the chromosome 10, this is 10 to the minus 353. So we did a small GWAS study for the Japanese populations, and we didn't see any complement factors. We only saw the chromosome 10, ARMS2 to HTRA1 locus. So we looked into the sequence of the AMD patient, 226 controls versus 228. We saw big deletions in this region. What does this mean for the AMD patient? We took this promoter regions and then we hook it up to luciferase assay. You can see that if you have a deletion, patient with the deletion will have an increase of HTRA1 in the neuronal cell line. But in RP, it's the same. What does that mean? So once again, we made an iPS cell from this patient. You can see that the HTRA1 transcription level is the highest in the AMD patients. So I think that the HTRA1 induction is extremely important for the AMD onset. So what's happening here is that in normal sequence, you have a repressor in the promoter region that is binding, trying to suppress the transcription of the HTRA1. However, if you have a AMD sequence, you have a suppressor here. However, you will have a activator activating. This is another gain of function for the AMD patients. So what are these proteins? So I will not go through in details, but you can read our paper in JBC last month that we have identified the proteins where that is increasing. So what happens to if you increase the HTRA1? Well, we made a transgenic mice with the HTRA1 hooked up to a promoter to express in the entire mouse, and sure enough, if you overexpress the HTRA1, you will have a neovascularization, choroidal neovascularization in the mouse. This is the first choroidal neovascularization genetically modified mouse in the world, and then it's OCT, FA, and the ICG. So another development in the replacement therapy, and I think you have already heard from this Japanese group in RICAN that Masayo Takahashi has made a RP sheet from the patient, and then she has implanted this sheet for one woman last year, and then probably she will be reporting this this year. Also, Yoshiki Sasai, who has developed the eye cup for the world's first eye cup from ES cells. Unfortunately, he passed away last year, so Masayo Takahashi is now using this technology, but using the iPS cells to make an artificial eye cup for the humans, and then eventually transplant into the patients. And this is probably the final slide I will have, is that there's an editing tool called the zinc finger nuclease, TALEN, and CRISPR. These are the most advanced technology to cut and replace with the sequence that you choose. And in combination with this, you can isolate the patient iPS, you use this technology, replace with the correct genes, and then make a eye cup or a cells and transplant to the patient. So these are the things that I have been talking about: the global collection of collection of data, and also new technology about the testing and the drug development using the iPS cells, and gene and cell therapy of the eye genetic diseases, such as gene therapy and transplantations, and then also I didn't talk about artificial eye. So thank you for your attention.